This is a production of Cornell University.
Yeah, so this is a campaign stop for me, trying to get your vote to renew my joint appointment in plant breeding. So all of you plant breeding faculty, vote early and often, because when we vote, we win. Okay, so I'm going to really give two seminars in one today, and they are both chapters from George Stack's dissertation, which he completed last year, and also a lot of work on the powdery mildew project from Ali Kalla, a PhD student in Chris Smart's lab. And yeah, I will go ahead and recognize my collaborators here because many of them, some of them are in the room. So Stephen Snyder, you're going to see some beautiful SEM that he did in Joss Rose's lab. I have collaborated with Ginny Moore's lab for quite a while now. And also we were fortunate to have to be able to work with Ping Wong in entomology in Geneva. You'll see those data. And I was on sabbatical at Colorado State for a year and got to know John McKay very well. And he was very generous to share a segregating population with us. That's the heart of the second half of my talk. And we got cannabinoids from Nick Jackowitz at Serona Labs, a startup company that was based in Geneva. So I'm going to talk about hemp today, one of the 5 crops that we breed in the lab. And hemp is a very interesting crop because it has multiple market classes. There was a 1937 Popular Mechanics article that came out saying that hemp would become a billion-dollar industry, that Henry Ford built a car entirely made of hemp, and that we can make 25,000 products out of hemp. And that's probably true, but not all of those products are viable in the marketplace or competitive for price in the marketplace. So hemp is an emerging crop because it was only truly legalized by the 2018 Farm Bill. Prior to that, it was considered a Schedule I narcotic like heroin. We initially had to have a DEA registration to be able to Schedule I DEA registration just to handle the seeds. But now it's totally legalized. And if you work on hemp, you better like roller coasters because it has been a serious roller coaster through the CBD market. So that is our top market. Grown either in controlled environment settings or in high intensity horticultural settings. Right now, all of the interest is in fiber. And when we say fiber, we really mean either the long, strong bast fibers on the outside of the stem or the inner woody hurd. Those are 2 different product streams leading to 2 different markets. And we are going to see a real uptick in grain because the FDA recently approved the use of hemp seed meal as an animal feed ingredient for laying hens. So that is going to stimulate the grain market over the coming year. And actually, this bottom machine is actually a dual purpose harvester that can combine the grain from very tall hemp plants while also harvesting the stems for fiber products. So it is a diverse crop. There are cultivars that are bred for each of these different market classes. But still in New York, we only have less than 200 acres of hemp being grown. I grew 35 acres on our farm plots in Geneva this year, and that was about 1/3 of the acreage in New York State. So it is a very early emerging crop. But I think it has a lot of potential. We're waiting for the Farm Bill, the new Farm Bill, which hopefully will further relieve some of the regulatory burden on farmers. A lot of that is based on THC. THC, in many people's eyes, is viewed as a very bad thing. And so one of the goals in our breeding program is to breed for low or no THC to try to basically eliminate that risk for growers, no matter what the regulatory landscape is. We think there is a market for the protein in grain, whether it's for animal feed or for human food. For human food, we're looking at the functional properties of the seed storage proteins as well. But right now, it's all about fiber, mainly for building materials and technical products. And in order to separate that bast from the herd, that is a process called decortication. And we need to have varieties that are easy and efficient to decorticate and separate that long fiber from the herd. Hemp has been notoriously difficult to establish in direct seeded plantings. Germination is typically rarely over 50%. And there are very few herbicides that are registered for hemp. So early seedling establishment and early seedling vigor is a major trait right now to ensure farmer success. And as a part of that, one of my students here, Luis Monserrate, is particularly interested in breeding for large seed size. And our large seeded cultivars seem to help to solve that early establishment problem. And the other issue we've had is that all of the current hemp cultivars have been bred in Europe, pretty much, or Canada, and are not well adapted to southern latitudes. We have been bringing in material from tropical and subtropical China, which will address that issue. But all of that material tested hot. So these are our challenges in hemp breeding. We started from scratch in 2017. So where do we get germplasm? Fortunately, we have the USDA ARS Hemp Germplasm Repository in Geneva at Cornell AgriTech. And Zach Stansel, the curator there, has done a fantastic job of very rapidly accumulating a large assembly of germplasm, both working with foreign repositories and bringing that material to the US, but also working with folks to collect feral hemp from across North America. We've done a lot of work to better understand the genetics of THC production. I'll talk about some of that. We've done a lot to develop genetic mapping resources. I'm not going to talk about that too much today. One of our main focus right now is understanding the genes involved in flowering time. And again, maybe that's for the next talk. What I am going to talk about today is pest and disease resistance. And going to start out with powdery mildew. So this is a project that we did jointly between my lab and Chris Smart's lab. Powdery mildew is caused by the fungus Rollivinomyces ambrosiae. You can see some beautiful pictures here from Bill Weldon in David Gadori's lab. And it is a big problem in controlled environment production of hemp and cannabis. And in our climate in New York, we also see quite a bit of powdery mildew in CBD hemp grown in, again, in this intensive horticultural setting. So right from the outset in 2017, when we started growing hemp, we wanted to better understand the traits of the cultivars. I'll say that with air quotes, cultivars that were available. So we were very proactive at trying to reach out to everyone who had seeds and put those into small plot trials. And at that time, it was all about CBD. So these are pictures of our 2019 trials with 30 cultivars, one in Geneva, one at Bluegrass Lane. And these were a lot of work, a lot of cannabinoid analysis that Stephen did in Joss's lab. But you learn things by looking at all of this variety of germplasm. And one thing that we consistently saw was essentially 0 ratings for powdery mildew disease in this variety, FL58. So this is a graph of percent disease from ratings that were done by Ali Kalla from those trials. And you see we were getting pretty good levels of powdery mildew in some cultivars. But again, consistently, this cultivar FL58 was resistant. So when you look at the resistance mechanisms for powdery mildew, we see either R gene-based resistance, and there was a paper published in 2021 describing the first R gene from cannabis sativa. They called it PM1. We would expect this to be a dominant and race-specific mechanism. We don't know anything about the races of powdery mildew, hemp powdery mildew. So that's still an open book. I'm just going to say this has been an extremely frustrating situation because this gene was discovered in a high THC variety by a company called Dewey Scientific, a spinoff from Washington State University. Mark Lang started that company together with one of his students. You may know him. And they have refused to send that material to us. They claim that they've bred it into a hemp variety, so it should be compliant, but even still, they will not send it to us to look at and to compare with the resistance that we have in our hands. It's been extremely frustrating. I'm going to continue calling him out for this. Because I don't think it's right that they should publish this in a peer-reviewed paper and then not share the material. The other mechanism we can look at is a susceptibility gene-mediated mechanism. This should have broader spectrum resistance and would typically be recessive. And there was a nice paper published in 2021 by Pepin et al. Describing the MLO, mildew locus O gene family in cannabis sativa, with two strong genes as candidates for powdery mildew susceptibility. So we have here some frameworks for what we might expect in terms of mechanism of powdery mildew resistance in that FL58 cultivar. So we took that FL58 and crossed it with a susceptible line, one of our favorite varieties called TJ-CBD, pretty susceptible, and then looked at the F1, and the F1 had variable levels of susceptibility. We only looked at a small handful, but again, these are now AUDPC, area under the disease progress curve graphs, on the left from a misdrum inoculation that Ali did, and on the right from a field trial. Again, you can see FL58 resistant in the field trial and a range of susceptibilities across the F1. So that immediately sort of rules out that dominant R gene hypothesis, pretty much, I think, because we are seeing a range of susceptibilities. We picked two of those F1 individuals and self them. And hemp is dioecious, but you can trick a female plant to make pollen, allowing you to self-pollinate it. And generated two segregating F2 populations, both with about a little more than 350 individuals, and planted those out in a field trial in Geneva in 2021. And Ali, again, very rigorously surveyed this population to generate these very nice AUDPC curves. And I have shown the two different F2 populations here in different graphs where we see that 25% of the F2 are scored as resistant, essentially zero AUDPC, aligned with the FL58 parent. TJ's is again moderately susceptible, but across the other 75% of the population, we see a range of susceptibility. Okay, so that does fit this model of a single gene recessive susceptibility gene. So we genotyped those 700-ish F2 individuals. We used an Illumina array that was developed by Phylos Bioscience. Even though there were 55,000 SNPs, we only had 2,700 that were useful for George to build the linkage map. I'll just say these two individuals, we have now sequenced these and have high quality phase genome assemblies of them. FL58 is highly inbred and very homozygous, and TJCBD looks like it is a true F1 hybrid cultivar. So those were interesting choices in terms of the genomics, and George has a manuscript we're about to submit that describes some of the interesting comparative genomics that we're working on right now. Anyway, the Phylos array worked pretty well for us, spread across the genome, even though it was designed to hide THC lines. And when he did the QTL mapping, we saw a very highly significant single peak on chromosome 1. And then some other smaller minor peaks on chromosomes 2, 4, 8, and 9. So looking at that peak, again, we had pretty tight and strong support for that interval. And when you look at the genotypes and line them up with the phenotype, it does point to a recessive mechanism for a lack of susceptibility. While among the other, the heterozygous and homozygous susceptible allele, there is quite a range of AUDPCs across the rest of that material. So a pretty strong effect of this locus on chromosome 1. And lo and behold, within that, or very close to that confidence interval, is this clade5 CSMLO1 gene that was predicted by the Pepin paper as being a good candidate for susceptibility. Very close to our peak marker. So I mentioned we sequenced those two parents, FL58 and TJCBD, did PacBio sequencing and OmniC to get a nice phased assembly. And again, lo and behold, in the resistant FL58 cultivar, there is a large insertion in that MLO1 gene, about 6.8 kb. A portion of it looks like a transposable element, and that insertion does result in a differentially spliced transcript, which would have a premature stop codon and disrupt function of that susceptibility protein. The interesting thing, we were pretty concerned about this early on, and I'll show you in the next slide how we resolved this. When we started doing RT-PCR of this gene, we were also detecting a full-length open reading frame. So that caused us some concern. How can this cultivar be resistant if it's producing functional susceptibility protein? So we spent quite a bit of time working on this and did RT-PCR from both uninoculated and inoculated plants. So that's the open bar versus the hatched bar. But really, I want you to focus on the green bars, which is the proportion of transcript that has the premature stop codon, so the disrupted transcript. And when we looked at FL58, yes, we can detect a very small amount of that full-length transcript, but the vast majority of transcript that's being produced is the defective, the interrupted version of the gene. I'm going to show you on the next slide, we've developed some markers to detect that insertion in the MLO1 gene. And those markers pointed us to some other germplasm that we had, which was heterozygous. And so you can see again in a heterozygote for that insertion, we're seeing about half of the full-length transcript and about half of the transcript with the insertion. And at least in this line, we do see an upregulation upon inoculation of the expression of that MLO1 gene. So now we have a few good markers linked to that MLO1 allele that has the insertion, which is effectively a knockout of the susceptibility gene. We can now use those markers to intergress that locus into other elite germplasms. Both, we like to use paste markers. So we have a couple of different paste markers as well as gel-based markers where we can clearly see the segregation of that insertion. So that was a very nice story. It all wrapped up, everything makes sense. It all wrapped up into a very nice package as a chapter in George's dissertation. The next step, we do want to do additional, more detailed RNA-seq to look at the expression of that MLO1 gene and the other clade5 MLO gene, which is MLO4. My student Lou Vignale is working on all of these follow-up questions. We're very interested to see if it will confer resistance in other genetic backgrounds and whether that MLO1 knockout confers any debilitated phenotype, because that's often the case with these susceptibility genes. We have not seen that so far with FL58. And we're also very interested in the resistance mechanisms between hop powdery mildew and hemp powdery mildew, and whether we have resistance genes to those alternate pathogens, resistance to hop powdery mildew and hemp, or resistance to hemp powdery mildew and hop. So that's something that Lou is working on as well. So she has been integrating it into other backgrounds and making crosses to get it into the homozygous state. And we have also been working with the USDA germplasm repository to screen other material and search for other sources of powdery mildew resistance. Maybe we will find that PM1 gene in other genotypes and won't have to go back to Dewey Scientific to beg them again for that. I'll pause here. Any questions about the powdery mildew story before I move on to herbivores? Okay. So again, a benefit of doing all of these small plot variety trials, it was a lot of work. It's a pain in the butt, getting the seed from all these companies and doing all the phenotyping, is yet you do learn interesting things. And again, in 2019, we planted out all of these grain and fiber varieties in small plot trials. And the observation that George made was that some of them were really getting hammered, primarily by Japanese beetle, which is a pest that we have a lot of that they don't have a lot of in some other big hemp growing regions like Colorado and Oregon. They really preferred these 4 varieties shaded in blue, which happened to all come from the breeding program in Ukraine. And the other feature of those 4 is that they are all cannabinoid free. So that's a pretty strong correlation that the 4 cannabinoid free lines just get hammered by leaf feeding beetles, whereas pretty much everything else that's making cannabinoids has at best a low level of herbivory. There are reports in the literature that cannabinoids may have some effects on arthropod health and feeding. So again, I think we had some good hypotheses that we could test to see what the role is of cannabinoids on at least this type of leaf feeding herbivore. So I have to give you a short background on cannabinoid biochemistry. If you're a hemp or cannabis scientist, this is a lot of what we all think about is cannabinoids on the medicinal side. But here we're looking at these on the field crop side. So this is a very simple diagram of the biosynthetic pathway. Combine GPP and olivetolic acid to make what they call the mother of all cannabinoids, CBG. And then depending on which synthase enzyme is present in those trichomes, you will either use that CBG as a substrate to make CBD, or if you have THCA synthase, you can convert that CBG into THC. So again, depending on the combination or the expression of those two enzymes, we have different cannabinoid chemotypes as we describe them. If the plant lacks active CBDA synthase, it will only make THCA synthase. We call that chemotype 1. That is the adult use drug type. But you might have plants that are expressing each enzyme. And if you have both enzymes present in those trichomes, they will make about an equal ratio of CBD and THC. We call that chemotype 2. It is also non-compliant, so it's still not classified as hemp in that case. Hemp is chemotype 3, so it is not expressing any active THCA synthase. It only expresses active CBDA synthase, thus we get CBD. The twist here is that CBDA synthase is promiscuous and makes a little bit of THC in about a 25-to-1 ratio. So hemp does make THC, and if you do that calculation, when you get to about a 7 or 8% by dry weight CBD line, it will exceed the 0.3% THC threshold. Then it's no longer hemp. So as John McKay says, hemp is a phenotype. Think about that. We can also find lines where both enzymes are knocked out, and then we see the accumulation of the precursor. So CBG dominant lines are chemotype 4. And so we now know that those two synthase genes, CBDA synthase and THCA synthase, sit very close to each other on chromosome 7 at a region that has been named the B locus. We have not detected any recombination between those two genes, so we can effectively treat them as alleles and they display Mendelian inheritance. And we also have lines that, again, either have a knockout in the THCA synthase gene, we're calling that BT0 or BT null, a knockout in the CBDA synthase, we can call that BD null, and we also find lines that have the entire region deleted. So we call that the B null allele. So then you can look at these different allelic combinations. Again, typically hemp would be homozygous for the CBDA synthase, it would be chemotype 3. But if it was heterozygous for the active CBDA synthase and some knockout, it would also be chemotype 3. You have to be homozygous for that B null or one of those other knockouts to be chemotype 4. Okay. And then finally, we can also find knockouts in the upstream enzymes in the pathway. For example, knockouts in olivetolic acid synthase, which totally eliminate cannabinoids. So we're not making any CBG either, and that is chemotype 5, cannabinoid free. Okay, and that is determined again by a single locus that has been named the O locus. So if you're homozygous for the little O allele, you are cannabinoid free. If you're heterozygous, you make very low levels of cannabinoid, and you have to be homozygous for the big O allele of that olivetolic acid synthase to be truly cannabinoid competent. So John McKay's lab, Patrick Woods in his group, generated a mapping population by crossing one of these cannabinoid-free cultivars, USO 31 from Ukraine, with an Italian fiber variety, Carmignola, and generated a segregating F2 population, which he shared with us. And it turns out that F2 population was very nicely segregating. He used a single F1 and selfed that. So it was a true F2 population. And that F1 was heterozygous at both the B and the O loci. So we have very nice 9-to-4-to-3 segregation of cannabinoid competence and production of CBD versus CBG. So let me walk you through this. So this is a log scale of total cannabinoids. So high cannabinoid on the right and essentially cannabinoid free on the left. And that makes sense when you look at the little o homozygotes here. The light blue down to the light yellow are all in this cannabinoid free group. So those are the chemotype 5 plants. And then if you look across this line, the homozygous B null genotypes are going to be chemotype 4. They're going to make CBG. So the vertical scale is the proportion of CBG. So all those high CBG lines, the chemotype 4 lines are at the top, and the rest are the chemotype 3 CBD dominant lines. So that gave us a very nice segregating population to look at the role of cannabinoid competence. And we could also compare CBD versus CBG from this population. So George planted it out in paired field trials, one in Geneva, one in Ithaca in 2021. He rated herbivory weekly, and then generated what we call area under the herbivory progress curves. And we also measured foliar cannabinoids by HPLC. And Stephen again looked at differences in trichome density and morphology in a subset of these plants. This is a nice phenotype you like to see, right? I'm not a stats expert, but you don't really need stats when this is your result. Yeah, we do need stats, but I'll show you those next. So the plant on the right is chemotype 5, cannabinoid free, just got hammered by some leaf feeding herbivore, probably Japanese beetle, but it could have been slugs, it could have been something else. We're not entirely sure of that. And the cannabinoid competent CBD line on the left, relatively little herbivory. This is a graph on the scale of herbivore damage expressed as the log AUHPC plus 1. Okay, so keep that in mind. The bottom line is that we do see a significant difference in the area under the herbivory progress curve between the cannabinoid-free lines in this F2 population versus either the CBD dominant or CBG dominant lines. There was much more herbivory in Ithaca than there was in Geneva. That was a local environmental effect, but still significant difference between the cannabinoid-free and cannabinoid-competent lines. Again, here we're graphing the log of total cannabinoids, so high cannabinoid on the right, low cannabinoid on the left. And in both sites, we see a significant negative relationship between herbivory and cannabinoid concentration. But that's all in the field. Again, we didn't really know exactly who was chewing on those leaves. So we wanted to try to counter any criticism about that by bringing leaves inside and putting them in a no-choice bioassay. This is where we got the help from Ping Wang. He provided us cabbage looper from his colony, and George picked out 6 CBD dominant lines and 6 cannabinoid free lines, and did 3 reps of feeding with 1 cabbage looper per leaf, and then measured mortality, position on the leaf, area consumed, and final mass of that cabbage looper. So again, they perform much better on cannabinoid-free leaves, significantly more leaf area consumed of the cannabinoid-free lines, significantly greater mass of the cabbage looper on cannabinoid-free lines, and significantly, I think this is significantly greater mortality on the CBD leaves than on the cannabinoid-free leaves. So all of that fits. And the other interesting thing is that these cannabinoid-free lines, normally these sessile glandular trichomes are the site of cannabinoid biosynthesis. you can see in the cannabinoid competent leaves, they're very swollen. This interesting observation that Stephen made that they seem to be collapsed in the cannabinoid free line. Also that there's a much higher density of trichomes on the leaf on the abaxial side, on the bottom side than on the top side, the adaxial side. And when George looked at the position of those cabbage loopers, that fit, right? So they were much more likely to be on the abaxial side on the cannabinoid free lines than they were on the cannabinoid competent lines, because that's where all the CBD is on these chemotype 3 leaves. Okay, so again, just these are whole leaves. Maybe there were differences in the toughness of the leaf. When you talk to entomologists, right, they want to know what was the toughness of the leaf, what was the nitrogen content in the leaf, what was the water content. Okay, so we'll do artificial diet assays then, eliminating all of those sort of tissue effects. So we worked with this company, Serona Labs, who had a nice water-soluble cannabinoid emulsion, incorporated it into the insect diet, and George did both an incorporation formula and then also painted the cannabinoids on the surface of the diet. And again, the cabbage loopers grew much less on the diet with cannabinoids integrated into it. And there was a significant effect on final mass with higher levels of CBD and well, this is not significant, but numerically different. The surface bioassay actually worked a little bit better and gave us the ability to do a little more range of concentration. And we did see a dosage effect with only 0.01% CBD, no significant difference in percent survival. But if you go up to 0.1, significant decrease in survival, and we killed all the cabbage loopers with 1% CBD, and most of them with 1% CBG. So all of those data fit the story, leading us to this conclusion that herbivory damage is related to cannabinoid concentration, that our cabbage loopers perform better. So it wasn't just something happening in the field. It wasn't something about leaf toughness, because we also saw the same effect with artificial diets. And we came to the conclusion that probably breeding cannabinoid-free cultivars is a bad idea, not a good idea. But we can breed for high CBG lines, this chemotype 4, which produce essentially no THC. So they will be essentially cannabinoid-free, THC-free, but not cannabinoid-free, and should still be able to defend themselves against herbivores. Now I'm showing a picture of a corn earworm because corn earworm goes right for the high CBD flowers. So the hypothesis there is that corn earworm don't care about CBD because that's where we find them in the plant. Same thing with cannabis aphid. So cannabis aphid is very abundant in the top parts of the CBD flower at the end of the season. So I'm not going to say that this is a silver bullet for all insect problems, but it does seem to have some effect on those leaf-feeding herbivores. So I will wrap things up. I need to thank the very generous funding we've gotten from New York State primarily through Ag and Markets, but also through Office of Cannabis Management and Empire State Development. We have had grants from industry, and this project on powdery mildew was funded by FFAR in cooperation with Scott's. And please email me or check out our website if you have any other questions. So I would be happy to take questions now.
About 10 minutes, Brill, Larry. to see how we are going to vote as a faculty.
Yeah.
First question from a graduate student or Abe's. Great to see you. Any graduate student? Yes.
I was wondering about the SGs, like there's trade-offs involved in knocking that out.
Yeah, so that is often by knocking out the susceptibility gene, you see an impact on vigor in other systems, and we have not seen that. We don't really know what the function of those S gene proteins is. Yeah, it's still an open question.
Sam to choose from. Sam in the back and Sam in front.
Would you ever foresee a type of hemp that combines, talk about those different functions that you could or how to give fiber or grain? Would you ever see a variety that combines those two or no, but then it would extend?
Yeah, so there are some very mature hemp industries in Europe and in China. And I think we in the US are only just beginning to learn how those mature industries are performing. I was just at a conference where I heard a talk from the president of the largest hemp company in the world in China. And they grow hemp in a very specific way and harvest it and process it in a very specific way to get textile grade fiber. It doesn't go to seed, so no, they're not going to get textile grade fiber and grain from the same plant. And they don't get any herd either because they're harvesting before the accumulation of any herd. In contrast, there is a mature grain industry in Canada, in Manitoba, Alberta, Saskatchewan, and they have never collected any of the straw. Their crop grows about 4 feet tall. They combine it with their big, huge, wide combines. And they are the largest producer of hemp grain. You could buy it in the store from Manitoba Harvest, called Hemp Hearts. So those are two mature hemp industries that are single purpose. So whether we can develop multi-purpose crops that are profitable and actually meet the needs of the end users, we do the economic modeling of that and it comes out beneficial, but what I understand right now is that the end users are not going to want those products from dual purpose crops.
I think I had tried this in parts. I visited you that one time. Tasty.
High in protein.
Yeah.
You might get 3 in omega-6 fatty acid. Chickens love them. Yeah, so sorry, I should be repeating the question. So the question is, there are only about 200 acres of hemp. I don't know the exact numbers of how many acres of cannabis there are. I would guess, I'm going to guess somewhere probably around 300 or 400 acres of outdoor grown cannabis. So what is our outreach strategy? My outreach strategy is to provide knowledge to growers and allow them to make the choice of whether they want to grow hemp. There has to be a market first, and we do not have any hemp processors in New York State. Again, I have a decorticator, and I am the only one in the Northeast with a hemp decorticator. So we need some investment in hemp processing before I would ever suggest a hemp farmer to plant the hemp. But I think now we have delivered field days every year since 2017. The best attended one had about 260 people. This year we had about 150 people. So there is definitely interest, and we show people how to grow hemp effectively for fiber grain and CBD. We just need the processing infrastructure And we need less regulation. That's also true. Okay, so we had a lot of CBD hemp growers in 2019. We grew about 2,000 or 2,500 acres of CBD hemp. The market crashed, but then New York State, this was the only state in the country that did this, authorized our experienced hemp growers to then get a cannabis grower's license. So everywhere else in the country, you cannot have both licenses. You can't grow hemp and cannabis on the same farm. So New York was pretty unique in saying, okay, hemp growers, you can now start growing cannabis, go for it. And so all of my clientele then started growing cannabis pretty much. And we are not allowed to provide extension to still Schedule I drug producers.
All right, just a quick question for the New York State Office of Cannabis. Do they oversee hemp as well?
So they do oversee not the licensing, but the product side of CBD.
Oh.
And sales of CBD.
Years ago, at the height of the fervor of CBD marketing, from beginning to $7 correctly identified it as both sequencing syrup. What does it look like now in terms of CBD demand? I mean, is it flatline, is it still crashing?
Yeah, so the question is for me to describe the roller coaster of the CBD industry. Yeah, so again, hemp, people started being able to grow hemp in after the 2014 Farm Bill, and states had to pass their pilot program. Every state was different, but it really was the 2018 Farm Bill. So some early states, Kentucky, Colorado, Oregon, got started ahead of everyone else. We got started, our plan was passed in late 2016. We started planting grain and fiber hemp initially, but then in 2018, it became clear that CBD was going to be the thing. And 2019, after the 2018 Farm Bill passed, people went berserk. I'll give you an example. There is probably the best seed producer for CBD hemp was a company in Oregon called Oregon CBD. And estimates I've heard is that they sold 300 million seeds in 2019 at $1 a piece. $1 a piece.
Wow, you're in the right business.
So the owner, Seth Crawford of Oregon CBD, is a very clever guy, very scientifically oriented. He was a member of the FFAR Hemp Research Consortium. He bought a PacBio sequencer and started sequencing genomes, working with Kelly Vining at Oregon State and Todd Michael at Salk. And they have now leveraged all of those data into a pangenome. So we now have 75 phased genomes from that era, thanks to the estimated $300 million that he made in 2019. People were paying, if you're planting 2,500 plants an acre, you're paying $2,500 just for the seed per acre. And some people were planting 40 acres. There were 60,000 acres in the state of Oregon in 2019. Based on all that overproduction, processors, we went and visited Charlotte's Web, a company in Colorado, when we were on sabbatical. They were still processing hemp that was grown in 2019 in 2023. So what we're being told is that the processors have finally gotten through all of the backlog of biomass from 2019. And there is now finally an uptick on the grower side for the price of CBD hemp, and people are actually planting CBD hemp again. But now the market is so thinned out that really the only survivors are companies that are vertically integrated, that have a solid seed supplier, grow their own hemp, process it, and market it under their own brand. We have a couple of very good ones in New York. Beacon Skiff near Syracuse. They have so many different companies, right? They have fantastic apple orchard where the kids go and pick apples and pumpkins and things. And then they have a fantastic cannabis company on the farm
Plain land for the whole family.
for the whole family.
Yeah.
They make cider, and it's an incredible company. So they are a survivor and a success story. But very few other success stories in that industry.
We'll probably close here, but just to remind people, as Theron had said, there's a meeting at 5 p.m. on which room was it? Bradfield 1102.
There's one question on the chat from Syria. Any idea whether spotted lanternfly feeds on hemp? And he wonders if there's a similar story as cabbage looper. So that's an excellent question. We also share that same question for hop. So spotted lanternfly is coming in from the south, is a big threat to our vineyards for sure. But we have not seen damage on hop, and we have not seen damage on hemp so far. So yeah, we need to wait until the populations are bigger for sure, but at least for now, it does not seem to be a huge threat specifically to hemp.
Great, let's thank Larry.
This has been a production of Cornell University, on the web at cornell.edu.